Afternoon, everyone. Welcome to our December lecture. And there's a lot of exciting things today besides Dr. Bruno, and that is that we're introducing new members. And that's kind of where we're beginning. I'd like to welcome Dr. Helen Malone to the podium. She is the Vice Provost for Academic Policy and Faculty Resources. She is our person. She's the one that makes it work. Helen?
Thank you, everybody, for joining today. Thank you for being members of the Emeritus Academy. Thank you for your service to the university. Every year I come and I go back and I look through the history of the Emeritus Academy, and I'm reminded that this group started in 2014 as a way to recognize and acknowledge the work of our emeritus faculty and to allow space and time and room for you all to continue the great work that you did in your careers as faculty at the university, but now to continue that work and have that opportunity to continue and to have this group of colleagues. For that work, I'm grateful, the Office of Academic Affairs is grateful, and the Provost is grateful. We're glad to have you continuing to work. We're glad to be able to provide some support to this group to continue to meet together and share your scholarship and your work with one another, and to continue to do your own work in your own space with the grants that we offer. So I love coming every year to hand out the new membership certificate. So I think without further ado, our Dean, I'll welcome you back and you can get us started. So congratulations in advance. I'll be here.
I think this hand mic is turned off, so we shouldn't get any feedback. Okay. Our new members for this fall in alphabetical order, and I'm starting with the folks that are here today. Professor Philip Binkley. He's from the College of Medicine and Public Health. Our next person is Nina Couch. She is from the University Library System, but has quite an interesting research set of projects with dance. Our next person, Avner Friedman, from the Department of Mathematics. The next person is Deborah Larson from the School of Health and Rehab Sciences in the College of Medicine. Welcome. And Danielle, I'm stumbling over your name. Danielle Marks. Please welcome. She is from the Department of French in the College of Arts and Sciences. Dale Svensson from the Department of Psychology, College of Medicine. Oh, sorry. And the last person supposedly that's in the room is Kenneth Yeager from the Department of Psychology. No. Psychiatry. Yes, okay, sorry. College of Medicine. Are there any other new members that have made it here to the group? Seeing none? Yes? Look. Oh, really? Please come up and shake Helen's hand. We don't have a certificate for you because we didn't know you were going to be here. That does the in-person. We have 3 people online. Elizabeth Allen from the Department of Clinical Pediatrics. I'm not sure where she is. Hopefully she's there somewhere. Camilla Curran from the Department of Internal Medicine, Pediatrics. Hi. And Margaret Wazinski. Ordering her last name, I'm very sorry for that. Department of Arts Administration, Education and Policy, College of Arts and Sciences. All right, so I would like to move on to our lecture for today. Dr. John Bruno is from biology, chemistry, psychology is his background. He came to us in 1986 and retired in 2018 as a distinguished professor and chair from the Department of Psychology. The research has been very productive. He has authored 225 peer review articles, PI on extramural funding during each of his 32 years at OSU. That's quite a record. Been awarded the Teaching University Award, founding director of a very popular interdisciplinary undergrad major in neuroscience, and also a recipient of the University Service Award. His research focus resides within the interdisciplinary intersection of behavior, cognitive neuroscience, neuropharmacology, biological psychiatry. Wow, what a mouthful, huh? His talk today focuses on central issues in that space and is titled Neuroscience of Cognitive Control in Health and in Psychiatric Disorders. Please welcome Dr. Bruno.
Thank you very much for coming today in person. I'd like to thank the people who are out in Zoom land. I come back home to my academic home from Sarasota, Florida, where it's a little bit warmer. And so I may shiver a little bit through this talk, but it's good to see old friends, and I'm happy to deliver this lecture to you. The topics that we'll discuss today are several fold, okay? First of all, I'm going to introduce you to a term in cognitive psychology, cognitive neuroscience, called cognitive control. We'll talk about what is cognitive control. We'll talk about how the brain mediates it. Where is it, where is the expression of cognitive control located in the brain? The answer to that question is it's extensively mediated by brain structures. We'll talk about the neurochemistry a little bit, and in particular, the role of acetylcholine and the role of glutamate, two neurotransmitters in the brain that are particularly interested in this cognitive event. And then part of my research over the years was to develop animal models of various neuropsychiatric disorders so that we could study the etiology of possibly the etiology of some of the disorders, and then use these models to predict new, more efficacious novel therapeutics. And so I will show you that the cognitive dysfunctions that we see in schizophrenia, for instance, can be quite effectively modeled in the rodent. And I will show you some of that modeling. But the bottom line prediction, the bottom line of our modeling is that if we have validated animal models, then they should show a convergence of results in the studies that we do on neurochemistry and behavior. And I'll show you that that's indeed the case. So cognitive control is a process that enables flexibility in, sorry, it's sort of hard to look straight up, by regulating three executive stages of information processing that you're familiar with at some level. One is the process of attention. The second is the process of working memory. And the third is the process of cognitive flexibility. And what cognitive control does is allows us to be flexible in our decision-making so that we can approach stimuli that are seen as positive, reinforcing, safe, and we can distance ourselves from stimuli that are threatening, etc. And cognitive flexibility, cognitive control gives us the opportunity to make these decisions. If you look at where it's represented in the brain, it's extensively represented, as I alluded to, both in cortical regions as well as subcortical regions, probably a dozen various structures. And what we've done over the years is to create a schematic that looks like this. Don't worry, we're not going to go into this in great detail, but I want to highlight a couple areas of the brain that are important for this mediation. First of all, I'll tell you that the structures are both cortically located as well as subcortically located. And there are reciprocal interactions between them. So you can take advantage of what we call in cognitive science, upstream regulation and downstream regulation. One of the two areas I'm going to talk a lot about is this area here in the temporal lobe, in the temporal lobe, which is called the ventral hippocampus. And the ventral hippocampus during development sends projections up into the cortex, into the prefrontal cortex, an area of executive decision-making, and subcortically into an area called the nucleus accumbens. And this is where the valence, if you will, of stimuli, are they positive, are they negative, are they life-threatening, how important are they? And then once that valence is tagged here, it goes into this machinery here, the basal forebrain, and back up into cortex. And that is a process that is involved with attention, okay, and selective attention and focused attention. So you have the ventral hippocampus controlling the development of two key areas in cognitive control. Now, a little bit about schizophrenia. I know that there's people in this room that know a great deal about schizophrenia. Essentially, it is a very debilitating thought disorder. By definition, it is a thought disorder, and it's characterized in a number of different ways. I don't want to say in the old days, but there are newer ideologies around this disease. But we used to talk about three clusters of symptoms characteristic of this family of disorders called schizophrenia. One are the positive symptoms, positive because these are symptoms that don't normally appear. And so their addition to the behavioral repertoire, cognitive repertoire, is positive by addition. And those are things like hallucinations, delusional thinking. Then there are negative symptoms, avolition, ambivalent affect, inappropriate affect, or emotionality. And then third, and this is what I'm going to focus on in the talk, is a set of disorders called cognitive disorders. And what we find is that the schizophrenic exhibits significant deficits in cognitive control or executive function. Now, why is that important? Because the cognitive deficits seem to be what we call core symptoms. These are primary symptoms driving this psychosis. They emerge in what's called the prodromal stage, And so these cognitive deficits are appearing even earlier than the other positive and negative symptoms that I mentioned to you, so they appear early. They're also exhibit a bit of a trait function. If you look at first-degree relatives who do not develop schizophrenia, they still exhibit some of the cognitive control deficits that their schizophrenic cousins or brothers or twins exhibit. And so that raises some interesting questions that we can talk about. Most importantly, the seriousness of the cognitive deficits seems to predict the course of outcome of the disorder. Okay, the more severe the cognitive deficits, the more severe the cognitive control impairments, the more severe and the worse the prognosis is. And so what we have is a set of symptoms, cognitive control symptoms, that are predictive of outcome. And while we do a fairly good job treating the schizophrenic, both pharmacologically as well as therapeutically, and mostly in terms of dealing with positive symptoms, a little bit better with negative symptoms. But in the cognitive domain, we have very little to offer the schizophrenic patients, all right? And so hear what I'm telling you. I'm saying this class of deficit is predictive of outcome, is a core symptom, and it's an unmet need. And so this is an important area for us to make some progress in, we think. So we're going to talk about the developmental models of schizophrenia. There are three of them I'm going to talk about, and they have to do with manipulations in rodents that affect the development of the hippocampus during early development, during very sensitive periods. And we'll talk about how hippocampal function can be disrupted in three different ways. And I'll show you those ways in a minute. And then again, the question that we are concerned with is, if we look at these three different models of schizophrenic cognitive deficits in the rodents, if they're all sharing some degree of validity, then different models might be expected to yield similar results for neurotransmitter release and tasks of cognitive flexibility. If you're on to something with these models, they share a construct and face validity, then they may have predictive validity as well. And this bottom sentence here is basically a statement that says that they should share a predictive validity. Now, some human data to show you, this is just a cartoon up here, but this is a cross section. The hippocampus lies in the temporal lobe, and it's sort of shaped like an Amon's horn. It's curved, and you can see the curvature here. And then if you take a cross section out of the CA1 region, I don't know that this pointer's working, of the CA1 region, and then stain these neurons with a high resolution stain, you're left with these two photomicrographs on the bottom here. So these are pictures of real tissue, all right? Real tissue of a patient with schizophrenia on your right, and an age-matched, quote unquote, normal control on the left. And the thing that struck people when this was discovered probably 35 years ago, the dark cell, those are neurons. They're called pyramidal neurons. If you look at their shape, you can see maybe why that term comes, where that term comes from. But these are pyramidal neurons that receive information from areas of the brain and then carry it to other target areas of the hippocampus. And what you see in CA1 in the normal individual is a fair amount of organization here. you can see that they tend to be, they have the one end, the dendritic field pointing upward in most cases. They occupy a tight layer within the hippocampus. And then if you look at the hippocampus in the schizophrenic, you see what has been referred to as a disorganization. And this you can see how they're not lined up tidily like this here. And they look more like this disorganized state. They're scattered across the area. They're oriented in very different ways. And that has implications because the way the neuron is oriented influences the kinds of neurons during development that will create synapses to talk with that neuron, and also the kinds of neurons it talks to. So in this helter-skelter case here, you have a hippocampus getting information from areas of the brain and from neurons in areas of the brain that it typically doesn't receive information from. And then it passes that information onto different targets, inappropriate targets. And so we'll talk a little bit about this disorganization. For the biologists in the room, this comes about as an error in migration. So it's the migration of the neurons during early stages of fetal development and early perinatal development, where this nice tidy package gets lined up. And there are mechanisms that control migration, and those mechanisms are dysfunctional in the schizophrenic. And what you get is migratory errors. So neurons basically in the wrong parking places, if you will. So what we did at first, remember this is the neural structure, the distributed system that is influencing cognitive control. What we did at first many years ago was a rather heavy-handed experiment. And what we did was we took out the ventral hippocampus. We lesioned it with chemicals, and at an early stage of development, which influences, as I said, the development of this structure and this structure. And we asked the question, what would happen to cognitive control or elements of cognitive control if we were to eliminate the hippocampus? I'll show you those results momentarily. Then we got a little classier. We got a little more sophisticated. We got another big grant, which allowed us to do those things. And what we did was we didn't eliminate the target region, the hippocampus, but we discovered a way to turn the lights out. All right. And this molecule here, some of the biologists again will recognize this acronym. This is tetrodotoxin. What it does is inhibit sodium gates. And so it makes it hard for action potentials to go down the neuron. So it temporarily inhibits neurotransmission, or at least sodium calcium-based neurotransmission. And if you inject TTX into this ventral hippocampus, you can basically shut the hippocampus down for about 10 to 12 to 15 hours, right? We thought that this was a more interesting approach, doing it at various stages of neurodevelopment, where we can bring it back online 10 to 15 hours later. But if we administered the TTX at a time where we knew important communication was happening here, then we would use it as a way to manipulate the development of this whole system. And then the third, those are the question marks that we answered. If the third one is the introduction of a molecule that is receiving quite a bit of attention these days. It's a molecule called cholinergic acid. It's an endogenous molecule. Everybody in this room has cholinergic acid. It's made not in neurons like neurotransmitters, but it's made in a class of supporting cells called astrocytes, okay, which are glial cells within the brain. But this cholinergic acid has two pharmacological properties that are of great interest to us. One is they inhibit cholinergic transmission, and it inhibits or competes for with glutamate transmission. All right, and so you'll recall one of the sets of experiments I'm going to show you is what's going on with cholinergic and glutamatergic transmission in the brains of these models, and does it resemble what's happening in the brains of schizophrenia? You can see here, if you look just at these two parts of the graph here, one is in, again, the prefrontal cortex, and again, in the nucleus accumbens. And what you see relative to control patients in the open bar, the filled bar is schizophrenic patients. And there is a significant increase in the levels of cholinergic acid in these two critical brain structures in people diagnosed with schizophrenia. All right. So this is a problem. If the cognitive deficits are emerging because cholinergic and glutamatergic transmission is not what it should be, and you've got this distributed neural system floating around in a medium of a molecule cholinergic acid that further retards the ability of glutamate and acetylcholine to activate the nervous system, then you have some problems. And I suspect that molecules that are designed to control the synthesis and expression of this cholinergic acid are going to represent the next wave of pharmacological intervention in treating schizophrenia. So we develop models based on all three. Removal of the hippocampus. We lesioned it at various stages of development, turning the lights out in the hippocampus, shutting critical information down for periods of time, and then bringing it back online again. And then third, we took pregnant rats, and we fed those pregnant rats with the precursor of this cholinergic acid. It's called kynurenine. And so you can artificially elevate the levels of cholinergic acids in your experimental subjects in much the same way you see them elevated in the schizophrenic brain. So what are the predictions from these studies? The predictions are regulations in subcortical control of acetylcholine and glutamate release into the prefrontal cortex. These should be dysregulated because the hippocampus set that tone for release. Secondly, we believe that this dysregulation of acetylcholine and glutamate release is at the heart of some of the cognitive deficits that we see. And so I'll show you evidence that that is the case. Third, this dysregulation in cortical transmission and cognition are age dependent, okay? By age dependent, I mean that the symptoms that you see following brain damage, and this has been demonstrated in a variety of neurological conditions, depend upon the age at the time of the damage. If you think about the brain being developing over time and important processes taking place at various points in time, and you start manipulating, if you start manipulating this hippocampus at a time when the neurons from the hippocampus are growing up into the prefrontal cortex, and you interrupt that growth, that infiltration, you'll get one set of data. If you wait until the hippocampus in the adult, if you wait until the hippocampus is fully formed, and you go in and shut the lights out for 10 hours, or you remove it, you'll get a different set of data. And much of our thinking about the origins of certain neuropsychiatric diseases, particularly those that have a developmental component, is that there are events that happen at critical periods of development that have tremendous impact on the ultimate development of the nervous system. And if you make your manipulations during those sensitive periods, then the brain is miswired and neurons are misoriented and function that requires upon the timing and the expression of neuron A to neuron B, that kind of information is lost, leading to some of the deficits. So, turning to behavior, okay, how do we measure some of these things? There's an animal model of cognitive flexibility, cognitive control called set shifting. And this little guy here and his brothers and sisters are real good at it, okay, as we are as well. And for the psychiatrists in the room, the task I'm about to show you is analogous to the Wisconsin card sort. Okay, and so what we know is that it's a measure of cognitive flexibility. It's asking the rat to pay attention to shifting perceptual demands of the task. Okay, and I'll make those clear in a minute. And then to make its decision on the basis of the shifts in those perceptual elements. We know for a fact that the performance in this task relies upon the integrity of the prefrontal cortex. So the prefrontal cortex, if it's disrupted, maybe because the hippocampal neurons aren't telling it, showing it how to normally develop, then you'd expect some problems with a task like this. Performance in this task is also highly dependent. Many studies from our laboratory and others have demonstrated that if cholinergic transmission, glutamatergic transmission within the prefrontal cortex are impaired, you do poorly in this task. All right? So, performance of this task in other ways, like the Wisconsin card sort or other set shifting tasks. Schizophrenics, the family of schizophrenic disorders, show clear impairments in these tasks, okay? And the kinds of impairments that the schizophrenic exhibits in this task are the very kinds of impairments that the three models of cognitive flexibility that I talked about. There are different stages of the task. One, and I'll define these for you momentarily. One is the extra dimensional shift, and the other is reversal stages. And so what are the data? This is a high test graphic that, yes, sir. When no one's yes, it's a very good question. And I don't mean to be cavalier about it. I did not say that we developed schizophrenia in a mouse, though for all I know, all mice are schizophrenic. But what we've done is developed by manipulating the same kind of biology that is impaired that we know is impaired in the schizophrenic brain. We predict and demonstrate that we get cognitive dysfunctions that resemble those seen in schizophrenia. So what we're doing is modeling the cognitive dysfunctions in schizophrenia by reproducing the pathology in the rodent brain. That distinction clear to you, sir? Yeah. Okay, so the key to this task is that there are two bowls that the rat gets exposed to, and they're trained here. And you can see what the reward in this case is a little piece of cereal that is hidden under one of these two bowls. But there are three sets of cues that the animal uses to solve this puzzle. There is, you can see the purple texture around the bowls, so the rat attends to that. In this case, they're the same. Then the cereal pellets are buried underneath this digging material here. And the digging material can be of different kinds of material. It can be dense material, it can be made of wood, it can be made of metal, it can be made of any number of things. And then the digging material itself is scented, okay, scented with, in this case, rose, in this case, gardenia, all right? And so what the rat and the human have to do is pay attention to each of these three perceptions, these stimuli, visual, tactile, olfactory. And what we do in this task is we train animals to say, we teach them that the olfactory cue is going to be critical. And you can disregard the other two. And so they learn that if they go to the side that has the rose scent, they will get rewarded with the cereal. If they go to the gardenia, they will not get any reward. So it's a two-stage condition task, termination task. Then what you do without the rat knowing, or without the patient knowing in the Wisconsin card sort, is you change the rules of the game on. No longer is odor the key element, but texture is, right? So they keep going to the rose. They don't find the cereal under the rose anymore, okay? And they then start learning that the texture of this digging material is really predictive of where the cereal reward is. And when you think about how we make decisions, this is in keeping with how we make decisions too, because if we're good decision makers, what we do is we remain very attentive to the cues that we get from our environment, our history of reinforcement. As reinforcement goes down, we're probably not doing things right. As reinforcement goes up, we tend to pay attention more to those kinds of events. So learning that odor predicted the cereal is called a set. And then when I switch it and turn it to the texture of the digging material predictive of the cereal, that's called a set shift. Right? And so we look at rats in their set shifting behavior. He comes, he goes over to gardenia. In this case, the gardenia has the cereal. So a Fruit Loop cereal. One of the reasons I had to stay funded for 32 years was that I went through a lot of Fruit Loops. Actually, my graduate students went through a lot of Fruit Loops, I think. But when you look at the task, I want to call your attention to these are different stages of learning this task. All right, and what we're going to focus on are reversal 1 and the ED, the extra dimensional shift. All right. These are terms that come out of the clinical literature, all right? And what we find is that individuals with prefrontal damage, like schizophrenics, have trouble making a reversal, okay? And a reversal is, let's say, rose was associated with the cereal and gardenia wasn't. Then all of a sudden you change it up, and now you've switched the positive stimulus and the negative stimulus. And so now the gardenia is associated with the cereal and the rose isn't. That's called a reversal. All right? And we do reversals all the time. We learn reversals. And you need to have the prefrontal cortex in order to make an effective reversal. The other most complex part of this task is what's called the extra-dimensional shift. And that's when you ask the animal to shift his search strategy from one set, namely odor, to another set, namely texture. All right? That takes a while to learn. And so you can see here, these are two of the models that we've been talking about. One is raising the cholinergic acid level. That's on the left side of this graph, okay? So we have controls, and then during early development, actually prenatal development, we raise the cholinergic acid levels on the animals. That's one of the models. And the second model is the turning the lights out. And what we do is we administer that TTX at postnatal day 7. And what's critical about postnatal day 7 is it's between days 10 and 12 that the neurons coming from the hippocampus arrive up to the prefrontal cortex. So what we're doing is turning the lights out while those neurons are on their way, navigating up to the prefrontal cortex. And what that does is causes a migrational error, okay? But what I want you to gather from this is that the very two stages of this task, the reversal stage and the extra-dimensional shift stage, are the ones where the model of schizophrenia here, elevated cholinergic acid, and the model of schizophrenia here, X treatment at postnatal day 10 or day 7, produce similar kinds of deficits. And these kinds of deficits, if I were to go into the clinic and repeat this task, these are the same kinds of deficits that the schizophrenic patient would show me. All right? So we were impressed by these data. Now, remember we talked about age dependency. It should matter when you have your brain damage, all right? Sometimes having brain damage early on is met with less deleterious consequences than having brain damage later on, okay? Other times, like in the field of teratology, we know that having exposure to teratogens early in development is worse than having exposure to teratogens later in development. So when you make the manipulation matters, all right? Age dependency is a key in our theoretical base. And so here we did the same thing with TTX that I showed you when you gave TTX at PD7, postnatal day 7, you got these big schizophrenic-like deficits in set shifting. You do it at postnatal day 32, which is adolescence in the rat. When you do it at postnatal day 32, same TTX, you see no deficits at all. Hey, everybody is equivalent, and particularly in extra-dimensional shifting. All right, and so you've missed the boat. You've missed the time window when you go and give PD at so late a date as to 37. All right, so now let's talk about what the neurochemistry behind these kinds of set shifting behaviors looks like. Does it involve acetylcholine? Does it involve glutamate? The answer is it involves both of them. Okay, here again is a set shifting data slide showing the discrimination stages here. And what I want to focus on is the data that you've seen already is the blue graph, okay? That's the control. And then the red graph, that's the animal that got the TTX. So you can see, here's the deficit. It takes more trials to learn the task. It takes more trials to learn the task here. And in the extra-dimensional shift, you can see that the TTX animal is taking longer to learn the task than its control, all right? What we did was we took an experimental agent, SSR, which is for the chemists in the room, it's a partial agonist. It's an agonist for nicotinic receptors, which are cholinergic, and it's an agonist for the NMDA receptor, which is glutamatergic. And so what SSR does is to facilitate cholinergic and glutamatergic transmission. And we take that, and you can see that when you add that drug to the animal, you can see its reversal deficits go back down to control levels in the white bar. Its extra-dimensional shift data go down to control levels in the white bar. We've also done the same experiment with, not with an agonist, but with an antagonist. This drug here, some of you know, MK801, is an NMDA blocker. So it blocks glutamate transmission at the NMDA site. And now here, this is a blocker. The one before I showed you facilitated. It was an agonist. It facilitated transmission. And so here you can see best probably in this case here, and say, here's the control value. These are otherwise normal animals. You give them an NMDA blocker, MK801, and they show you the same kind of deficits that you see when you give cholinergic acid or TTX or anything like that. Last, test of the model, cholinergic acid. Remember, cholinergic acid gave you deficits in reversal 1 and extra-dimensional shifting. What we did was we treated with a compound called galantamine. And when we treated with galantamine, you could see that the reversal deficit in the cholinergic acid animal relative to control, and the reversal and the extra-dimensional shift deficit relative to control, those were both eliminated when you treated with galantamine. So when you blocked cholinergic acid's effects, you did not get the cognitive deficits. Okay, so do the models lead to activation in our models? This will be the last series of studies I'll show you. What we did was we went directly into the distributed circuit that I told you about. And what we did was we activated the nucleus accumbens with administration of a molecule called NMDA, okay? And NMDA is a glutamate agonist, and so it activates this area here, and then we eavesdropped with biosensors. We put biosensors into the prefrontal cortex, and we asked the question, what happens with the release of various neurotransmitters? Now, what would the prediction be? The prediction would be that in normal animals, if you had put NMDA into the shell of the accumbens, you would stimulate acetylcholine and glutamate in the prefrontal cortex. That's the way the anatomy works, all right? The question is, would it do the same thing in one of our 3 models? Okay. And focus your attention on the middle panel here, please, because I don't have time to go over these graphs in great detail. But the model here is the lesioning the hippocampus early on in neonatal time, where you disrupt the projections to the prefrontal cortex. We've been over that. So when you see that in control animals, the controls are these open boxes. When you deliver NMDA into the nucleus accumbens of the animal, you go from basal levels of acetylcholine here. All of a sudden, as soon as the drug hits the nucleus accumbens, you get a doubling of acetylcholine release in the prefrontal cortex. Remember, these are normal animals. And when you turn the NMDA off, acetylcholine levels go back down to baseline. So that shows the stimulation of acetylcholine release in normal animals. Now look at the black square. That's the animal that has the lesion, okay? You can see the black square, even though you give the NMDA, same doses and everything, into that brain, you get no bang for your buck. You get no stimulation of acetylcholine increase in the neonatal animal. So the model has sort of disconnected activity in the nucleus accumbens with activity in the prefrontal cortex. This goes to show you that if you do the same manipulation, but you do it in adults, not neonates, look what happens. The adult lesioned animals look just like the shams. Why is that? That's because the fibers have already gotten to the prefrontal cortex, because now you're in a fully mature system and you see different results there. This will summarize it a little better, folks. On the x-axis here, we have the dose of NMDA that we're putting down into the prefrontal, into the nucleus accumbens. And then we're measuring now, in this case, glutamate. In control animals, here, these are developmental controls. Here, you get a nice dose response of glutamate release. In the control animals here, again, nice dose response of glutamate release following NMDA. Look what happens to the lesioned animals. No effect. Modest effect. Little effect to the highest doses. So there's a subsensitivity or an insensitivity of cortical glutamate and acetylcholine transmission in these three animal models. Behavioral data in set shifting converges and shows great similarity in these three animal models. So in summary, cognitive control can be effectively modeled in rodents, all right? And notice, sir, that it doesn't say schizophrenia can be effectively produced in rodents. It says that cognitive control deficits can be effectively modeled in rodents. Second, dysfunctions of the ventral hippocampus develop in the development of the ventral hippocampus result in deficits in the subcortical activation of prefrontal acetylcholine and glutamate. These neurochemical deficits are accompanied by and appear to mediate the impaired performance in the behavioral task. Remember, we could stimulate acetylcholine release, the deficits went down. We could block glutamate release, the deficits went up. As postulated in studies of schizophrenia, age at the time of brain damage also markedly influences the probability of deficits. Okay, and so in closing, I think that one can model a variety of complex behaviors in our mammalian brothers and sisters, and then that the rodent is a good model for looking at this kind of set shifting cognitive flexibility. And it appears as if the distributed neural circuit and the neurochemistry that's released looks similar in the models as well as in the clinical condition. And so my last slide, it takes a village, right? This is my it takes a village slide. Over the years, I've had collaborations at each of those universities, each of those areas. Colleague Martin Sartor for years, He and I worked on these problems here at Ohio State. We work on them in Maryland, in Franklin, Rosalind Franklin, and University of Calgary in probably my favorite place to work on them. Feel good right about now. But I want to thank you for your attention, and I'll be happy to answer any questions you have.
Great, we have our first question right here.
So that was really fascinating. Thank you for making it very clear.
Thank you.
It was fairly easy to follow, at least for me. So I'm an MD. Maybe that makes a difference, right? But the question I have is what you're actually postulating here. You're postulating a biological cause of schizophrenia, and that postulate is that the cause is damage to the brain at an early age, although in humans we're talking now, although that age is unspecified. So could you expound on what the human theory of schizophrenia would be as implicated by this research?
This is, first of all, it addresses certain components of the information processing deficits that you see in schizophrenia. All right, so what we're doing is modeling attention and cognitive control. Okay, it says that early on, well, you know for as sure that the current thinking about the pathophysiology of schizophrenia is that many of the disruptions are taking place during the late second trimester and early third trimester. And so these are early events typically related to the orientation and migration of those neurons I talked to you about. So what I'm saying is one of the things that we know about schizophrenia is that the cortex, it's difficult to activate the cortex when the subcortex gets activated. Okay, and so the prefrontal cortical activity, if you do neuroimaging, if you do PET, what you'll see is a paucity of activity in the frontal lobe. And the reason that is the case is, and whether you're engaged in a task or not, you see a paucity of activity in the frontal lobe. So what I'm saying is that there are developmental periods, there are sensitive periods in development in the rodent that we can produce cognitive deficits that resemble the cognitive deficits that look like in schizophrenia. Bypass that window and make our manipulations later, after things have become wired, if you will, migration has taken place, we don't see those same kinds of deficits. So that's what we're trying to model. Now, there's a whole second part of this study that involves, it's a two-hit hypothesis, as many hypotheses are in psychiatry. So we think that this loss of subcortical ability to activate the cortex that happens early in the perinatal period. Then in the period of adolescence, there are stressors that come, and the only way to deal with those stressors is to be able to activate that system, and the system's not activated. And so that becomes the second hit, okay? And so, and what we find is that if we take animals like these three models, stress them during adolescence, and let them grow into adulthood, we find much more severe behavioral cognitive deficits, affective deficits than we do if we just have one stage of the model.
Thank you. I'm a cardiologist, so
Yes.
this is way out of my field, and so this is probably a.
For 1 second, please. There's a question behind
Oh,
you,
I'm
sir.
sorry. So this is way out of my field and probably fairly naive, but I'm thinking of the mathematician, John Nash, who developed schizophrenia, and he said that later in his life, he described it as being similar to dieting. He could recognize his delusions and hallucinations and put them aside. And I guess and function better. And I guess the question is, he seemed to have maybe developed a cortical means of counteracting these dysregulated signals from the hippocampus. Is there a neurochemical correlate of that, or over time, do some of these things like hyaluronic acid diminish in concentration and allow this to be reversed?
If you elevate chynurenic acid during the embryonic period, you get the deficient animal that I showed you. You elevate chynurenic acid after adolescence, don't get. John Nash was anything but normal, first of all. And what people often mistakenly do is think about intellectual deficits in schizophrenics, and very specific, oftentimes on generalized IQ tests, you won't see significant departures. Population. It's when you have tasks that target the ability to cortically activate the prefrontal and to have these executive functions. It's where you see the deficits are in attention, working memory, and in set shifting or cognitive flexibility. You see those deficits. And Nash, who was capable of many things, still revealed deficits in attentional processing.
Other questions, Dr. Friedman? Did you have that?
And identify associated with schizophrenia.
Yes, the molecular genetics work is exploded. The best way that I can specific genes, interestingly enough, the three most important gene operations that have been shown to be efficient or polymorphic in schizophrenia. One, that controls neuronal migration. Two, a gene that controls the nicotine acetylcholine receptor. Three, genes that regulate the transmission of dopamine, glutamate. So the genetics are consistent with the kinds of pressure points that I showed in this talk here. But the genetics, the best way I could summarize the genetics of schizophrenia is in the title of a talk, title of a paper of one of my colleagues. And it was entitled something to the effect of many genes doing little things. So finding a single gene product or gene polymorphism that leads complex as schizophrenia. But the four targets that I just mentioned to you, dopamine, migration, are all problems that are all built into our models. Good question, sir. Yes, sir.
Yeah, my question, I think, is somewhat related. I didn't hear the previous questions, but I heard your answer. But is schizophrenia, is it inherited disease? That's one question. Second question is that you talked about prenatal or fetal development. And I started thinking that premature babies, do they have an increased probability or increased chance to develop schizophrenia?
Three questions there, but I'll take them one at a time. the last one. Preemies there's a strong literature on executive function deficits in preemies or breech births. And so ability to focus and sustain attention is impaired in many cases of premature birth. And so executive functions there are. No evidence that I know of, strong evidence that I know of for birth delivery correlating with schizophrenia. But you had asked about the genetics of it, and the genetics or the concordance, the genetic loading in schizophrenia is quite high. And so first degree relatives have if one has schizophrenia, there's anywhere from 47 to 72% chance that the twin pair will have schizophrenia as well. So it is I would say a highly genetic. But when I say twin pairs, if one has it, the other one has a 50% chance of having it. That sounds like a big number, but there's 50% that don't get it. So it speaks to multi-factors, doesn't it? 50% is a high concordance ratio, but there are other things going on as well, highly genetically involved.
Dopamine blockers, and the cognitive effects are not really altered by them, and neither are the avolitional negative symptoms. And you also point out the multiple kinds of genes affecting different kinds of neurochemicals, etc. And it's a complex disorder, obviously, and yet it fits many of the things that we do know.
Thank you for your comment. It is the case that the negative symptoms have traditionally, in the early stages of dopamine antagonism, had gotten worse with therapy. Positive symptoms that were most respondent. Cognitive symptoms got worse. Then we went into typical antipsychotics, as you know, and first being probably clozapine. Didn't exacerbate negative symptoms and showed a bit of a bump in cognitive control.
And clozapine is not very strong as a dopamine. It's a dirty chemical. It's got a lot of other things going on.
It's a dirty chemical. Yes, it is.
Are there any other questions? Yes.
I know this is an interdisciplinary crowd, and I gave you an interdisciplinary talk. And I want to thank you for sticking around and going through the different levels of analysis.
So my question is, if you made a lot of nervous mice or hypervigilant mice, would they show some of these changes that you're seeing in what you're calling the schizophrenic mice?
Yeah, if you made them hypervigilant. Hypervigilant so that their attentional mechanisms were challenged.
So just scare the mice repeatedly, had a really bad life.
Yeah. I think that, attentional, mice are just a tough one to start with in the
Right.
first place because they're hypervigilant coming out of the gate. I think with rats, there's evidence that, that hypervigilance is a bit of a problem in these kinds of tasks, because what you've got to do is switch your attentional focus, all right? And if you're unable to divide your attentional resources properly, then you have difficulty making that cognitive switch. So hypervigilance and characteristic of a number of human disorders as well. But I would say, depending upon how you did it, if you did it, you would have problems with the kinds of tasks I'm talking about. Just like if you did gave a drug that interfered with working memory. Can't hold the rules of these tasks in mind as you're going through trial after trial, then performance goes way down. And so attention and working memory work together.
We would like to thank you so much for your lecture this afternoon.
Thank you. Thank you. Very, very patient. Thank you.